Not all drug-coated balloons are created equal. Um, every single technology has certain technological and biological principles. And drug-coated balloons, at the very beginning, essentially it started with the notion that a drug could be delivered in a single time fashion, that the drug would stay in the vessel wall, and that the drug would essentially exert a sustainable biological effect. So you can actually see the philosophy of the company or the innovators in every single design that is being created. Some technologies have actually gone with more crystalline coatings, which are actually less soluble. Therefore, they essentially provide a longer term release of the drug in the tissue. Actually, some went with lower concentrations of the drug and more soluble drugs that essentially allows the drug to disappear more quickly from the tissue. The reality is that essentially you need a drug to stay in the vessel wall, and the more crystalline you are, more drug you're going to have staying in the tissue over time. So when you actually see all the technologies with different excipients and different medications and different concentrations, despite the fact most of the companies are using nowadays paclitaxel, you see that the biological effects are actually quite different, which is very interesting in this area of local drug delivery. The IN.PACT drug-coated balloon is actually different than other technologies that followed a different technological principles. Particularly, actually, the IN.PACT DCB technology has what actually we call a crystalline coating, meaning essentially what happens is the paclitaxel and the what we call excipient, which is a carrier that takes paclitaxel into the vessel wall, that combination forms actually microcrystals. And these microcrystals, when essentially are attached to the vessel wall with the balloon, they stay there, they're covered with fibrin on the vessel wall, and this produce what we described long time ago as natural depots, long-term release related to the crystal. So this is the mechanism of action of drug-coated balloon, and nowadays we know that the solubility of the particle plays an important role in long-term effects. So the reality is when you decrease the size, the composition, or the solubility of this particle, you start actually seeing different type of pharmacokinetics and pharmacodynamics, and the biological result is going to change balloon to balloon. That is why, despite the fact, again, all these companies or different technologies use apparently the same medication, the biological effect, when you see the clinical data, is different. And because it has to do with solubility, sustainability of vascular effect, and residency of paclitaxel in tissue over time. So when we talk about dose, we have to be very careful because dose in drug-eluting stents is kind of in a way more predictable. It's like a pill that you take because you can essentially titrate the amount of drug inside of a polymer per millimeter square, and in a way kind of determine the way the medication is going to be released. In drug-coated balloons, we cannot really talk about dosing just because you claim you have X amount of drug on the surface of the balloon. The reason is because the final effective dose, or the dose that is finally delivered into the vessel wall, depends on many, many circumstances. It depends obviously on the concentration you have on the balloon, but also depends on the uniformity and durability of the coating. It depends on how much actually drug you're going to lose in transit. It all depends on how much drug you're going to lose during delivery. And it all depends about the adhesiveness of that coating on the vessel wall, and so forth and so forth, so forth. So I really think that this discussion about dosing, we have to be very careful when we frame it. And essentially, we have to frame it according to the solubility of the particle and sustainability of the vascular results. The sustainability of the vascular effect in local drug delivery depends on many, many factors. In order to be able to sustain a biological effect in restenosis, meaning prevent restenosis long term, it is absolutely crucial that we block the pro-inflammatory and the healing process that is actually leading to restenosis at the time in which is actually needed. That is why if you have a very short time of drug delivery, you may actually block inflammation and have an anti-restenotic effect at the very beginning. But if your drug delivery ends when the healing continues, you're going to see catch up over time. So that is why it's extremely important, and at the present time, especially in any type of local drug delivery, that the biological effect can be sustained over time. That is why coatings and formulations that maintain tissue levels over time, as long as actually possible, are going to have a better result at long term. If you see, for example, the data in a lot of the drug-coated balloons, you see that patency is achieved in a very interesting numbers. In almost all technologies around 1 year, but after 1 year, you start seeing a degradation of the effect over time. That is why it's important that that residency time gets to a point that blocks the anti-proliferative effect. So then once healing starts, then it's actually normal healing, no hyperproliferation of the vascular growth restenotic process. This is extremely important because local drug delivery in the peripheral arteries need to be progressive, need to be gradual, and need to be more prolonged than in the coronaries. If you look at experimental data, in 28 days of release, you actually get a sustainable vascular effect. In the peripheral vessels, perhaps you need that residency time to be a little bit longer because the volume, the amount of tissue, and the amount of plaque and the complexity of the disease. That is why, again, you see differences in clinical outcomes. And also that is why it is extremely important that that amount of drug that stays in the vessel wall stays as long as possible for any type of local drug delivery to work over time. I feel the field of local drug delivery in the peripheral field, changed our lives forever. So I really think that, we all need to be grateful that these technologies were aggressively pushed forward. And now we have at least therapies that can really help, critically ill patients.